Chapter 10, Photosynthesis. My introduction to photosynthesis is simply this. Life as we know it depends on photosynthesis. Photosynthesis is that all-exclusive link between light energy and chemical energy. This is what converts light energy into a form that we can use on Earth. If there were no photosynthesis, there wouldn't be life as we know it. We indirectly or directly eat plants for energy. And secondly, the air that we breathe, all of that comes from photosynthesis. So if you like eating, if you like breathing, you've got to love photosynthesis. In this chapter, you will learn how photosynthesis links life to the power of the sun, how light energy is converted into chemical energy, in steps designated as Photosystem 2 and 1. We will also learn how pigments are used to capture light energy and how this energy in the form of NAD and NADPH is transferred to the second step, the Calvin cycle. And in the Calvin cycle, sugar is made. And lastly, we'll talk about a couple of tricks that plants use to actually make an efficient use of photosynthesis, especially under stressful conditions. Okay, so what is photosynthesis? If I look at the word photosynthesis, it means to me light making, and in this case it's light making sugars. Isn't it true that we're going from CO2 to making sugar? So this is an anabolic process. We're going from small molecules to large, and for this reason we need a lot of energy. This is definitely an endergonic reaction, and all of this energy is coming from the sun. Now, this is just a side note, it won't be on the test, but there is a difference between autotrophs and heterotrophs. Autotrophs are self-feeders, and heterotrophs eat on others. We as humans are heterotrophs. We have to consume others so that we can stay alive. Autotrophs make their own food. That's the plant world, and algae and some blue-green bacteria, or the cyanobacteria. So they can make their own sugars, and that's what this chapter is all about. So what are the three major ingredients for photosynthesis? The three major ingredients are sunlight, carbon dioxide, and water. And as a result, we get sugar, glucose, and we get oxygen. So notice, if you take a look at the equations, isn't photosynthesis the opposite of cellular respiration? So if you break down sugar in a catabolic pathway, that's cellular respiration. But if you build up CO2, that is photosynthesis. So these two equations are opposites of each other. In figure 10.1, you can see the reaction of the three ingredients, CO2, water, and energy, and you can see here that it's ultimately going to make glucose and oxygen. Now, for photosynthesis to occur, we need a lot of energy. And energy, it moves around in the cell, as we learned before, in two ways. It either moves in the form of excited electrons, or it moves in the form of a phosphate group. So in this particular case, when we harvest light, it will be converted into both excited electrons and ATP. That's where the phosphate group is there's three phosphate groups in the tail of ATP. Now, where do the excited electrons come from? They come from the splitting of the water molecule, and it's sort of funny how this was found out. They weren't really sure where oxygen came from. Initially, many thought that oxygen came from the splitting of CO2. C, carbon, oxygen, and two oxygen. So the oxygen came from CO2. But then there's this group of bacteria that live in deep sea vents, and these are called purple sulfur bacteria. And actually, instead of splitting the water molecule, they actually split hydrogen sulfide. And believe it or not, instead of creating oxygen, they create sulfur. So from this rare group of bacteria that conduct anoxygenic photosynthesis, we know that the splitting of the water molecule produces excited, that's the source of electrons that will get excited in the photosynthesis process. So here it talks about the purple sulfur bacteria that I mentioned in the previous slide. So we know that oxygen comes from the splitting of water, and it does not come from CO2. So that's pretty interesting. And I promise you this will be a test question on the upcoming test. Okay, now there's two parts to photosynthesis. Remember, like in cellular respiration, there were 4 steps: glycolysis, pyruvate processing, the citric acid cycle, and then lastly, the electron transport chain. Well, photosynthesis only has 2 steps. It has the light capturing reactions, or the light dependent reactions. And then the second step is the Calvin cycle, or the light independent reactions. So notice here for light capturing reactions, this is where water is split, and this is where oxygen is formed. In the light capturing reactions, this is where we make a lot of energy. And remember, energy can move in 2 forms in cells. It can move as phosphate or it can move as electrons. And in these light capturing reactions, we're going to have both. Then the Calvin cycle is the step that produces sugar. And I always think Ca is for the Calvin cycle. Ca also is for carbs, carbohydrates. So this is where sugar is made. Notice CO2 has to come into the cycle, and then all of the energy, all of the electrons, and all of the ATP from the light capturing reactions go into the Calvin cycle to actually produce sugar in combination with CO2, carbon dioxide. Figure 10.2 puts everything together. We see sunlight at the top, then we see the light capturing reactions. And we notice here that water has to go in, and then this is where oxygen is produced. Why does water go in? It's because we don't need the oxygen, but we need the protons and we need the electrons to actually create ATP. Now we have a new electron carrier. Instead of NAD, as it was in cellular respiration, our electron carrier is NADP. And then the last step is the Calvin cycle. And in the Calvin cycle, all of that energy produced in the light capturing reactions is moved to the Calvin cycle along with CO2 to produce sugar. I really appreciate this figure because if you can put everything together, this is simply what photosynthesis is. Photosynthesis is 2 major steps to produce sugar. Okay, now where does photosynthesis occur? Photosynthesis occurs in the chloroplast. Chloroplast meaning green formed elements or bodies. Now, just like the mitochondria, there's an outer membrane and an inner membrane. The inner membrane is called thylakoids, and if you stack them up like pancakes, they're called grana. And then the liquid or space in between is called the stroma. Now, my face is covering the word stroma, but you need to know that for the exam. And in this diagram here, you can see the chloroplast within each of the cells found in the leaf. And luckily for you guys, when we did our microscopy lab and we looked at Alodia leaves, those leaves found in aquariums, then you could actually see a lot of beads inside the cell, and those are chloroplasts. This is where photosynthesis takes place. Both steps take place in the chloroplast. Now, how is light captured? Light is captured by pigments, and these pigments are hooked in to the inner membrane of the chloroplast. In other words, they're hooked in to the grana or the thylakoids. And the most common pigments usually found in most leaves are chlorophyll A and chlorophyll B. I guess you're getting the hang of it. Chloro, that prefix chloro is referring to green. So chlorophyll is green leaf pigments. So the funny thing about light is this, is in light is all colors are absorbed except for the color that you see. So you can see I'm wearing a blue shirt right now. And the reason why you can see that it's blue is because my shirt is absorbing all colors and reflecting blue. In the back you can see some pictures and you see that there's red, oranges, and yellows. Well, the reason why you can see red, oranges, and yellows is because the threads are absorbing all colors except for red, yellow, or orange, respectively. So now let's look at green plants. Why do we see them as green? Because they're absorbing all the light that there is in white light, which is the combination of all visible light, except for green, the reflecting green. Okay, so that brings us to electromagnetic radiation. And light is a form of energy, and that is this subject. We're taking light energy and we're converting it into chemical energy. Now, we can look at light in two ways. We can look at light as a wavelength, and we can also look at it as a little particle or a discrete package of light, and this is called a photon. So in the electromagnetic spectrum, we have varying degrees of wavelengths, and there's an inverse relationship with wavelength and energy. The shorter the wavelength, the greater the energy. The longer the wavelength, the shorter the energy, or the less energy it has. Now, we'll be talking about the visible light portion of light, and so this will be from 400 to 700 nanometers. So again, notice here at the very bottom of the slide, it says the energy of a photon of light is inversely proportional to its wavelength. So there's this opposite relationship between wavelength and energy. So shorter wavelengths have more energy than longer wavelengths. So this is the visible light spectrum, and thankfully we've done experiments in class. So at the 400 nanometer is blue light, and at the 700 N and the opposite end we have red and far red light. Photons may be absorbed, transmitted, or reflected when they strike an object. And remember, when they're reflected, that's what's giving them their color. So pigments absorb only certain wavelengths of light, and you can imagine it depends on the color of the pigment. Now, within a leaf, we only see basically green, but that doesn't mean that there aren't any other pigments or accessory pigments there in the leaf. So let's look at this diagram here. It looks like there's grass blades in the mortar and pestle, and what's being done, it's made into a mush, and they're probably using like an organic solvent, something like alcohol or ether. Then they're going to take this liquid on paper, and they're going to use the paper to actually sort out the pigments. And when you take a look at it, what looked initially totally green, we see that we have like xanthophyll, chlorophyll A and B, pheophytin, and carotene. So we have an array of pigments. We don't just have chlorophyll A and B that are green in color, but we can have other pigments that are often masked by the large quantities of chlorophyll A and B in the leaf. So there's two major classes of pigments. So the first class is chlorophyll, and they absorb red light and blue light, and I promise you this will be a test question. Then we have the carotenoids, and they absorb blue light and green light, so that makes sense, right? They absorb blue light and green light, but they reflect yellow, orange, and red, because the carotenoids are those colors, so that's how we can see that they exist. So the same with chlorophyll, they absorb all light except for green. We're going to see a diagram, and it's going to show us the action spectrum and the absorption spectrum. So this takes into account different wavelengths, the major ones chlorophyll A and B, and it also takes into account the inclusion of accessory pigments. So we know it says here wavelengths drive the live capturing reactions. And isn't that is what's happening in the light capturing, is that we're converting light energy into chemical energy. So the action spectrum tells us what wavelengths, and the absorption spectrum tells us how wavelengths of photons influence the amount of light absorbed by pigments. I love this classic but elegant experiment. They take a strand of algae, and algae is found typically in pond water or surface waters, and what they did is they shined light. You can almost see that it's white light, it's just regular light that's being shined through a prism, and the light looks like a rainbow because the prism is fracturing the light into different wavelengths. And you'll notice that oxygen is being formed where there's blue light and there's red light, and hardly any in the middle where there's green light. So this proves that most plants love blue light and red light. If we put all of this together, we have this absorption spectra. So notice here we have chlorophyll A and B, the green ones. They're in light green and dark green. And then you have some orange lines there representing the carotenoids. So if you look over the visible spectrum, which is the x-axis from 400 to 700 nanometers, you see that most of the pigments are picking up colors at either end of the visible light spectrum. The 400s, which is the blue, and the 700s, which is the red. Chlorophyll A and B are similar in structure. They have this long isoprenoid tail that hooks into the grana, or the thylakoids, the inner membranes of the chloroplast. And then the head looks like a great big radar, a cup, and it is a large ring structure that has magnesium in the middle. I'm just reminding you what isoprenoid means. It's a carbon-hydrogen, chain. So here we see chlorophyll A and chlorophyll B. And notice here you see a long tail of carbons and hydrogens. That's the isoprenoid tail. And then we see the left-hand side, which is a lot of ring structures together with magnesium in the center. It's sort of like a cup, and it's, facing the sun. Like, ooh, like where's the light so I can capture it? This ball and stick model shows you how actually chlorophyll looks like. So notice you have the tail that hooks into the membrane, and then you have the other end, which looks like it's capturing light. Form and function go hand in hand. We also have accessory pigments. What are accessory pigments? These are not the main pigments, but they're helping, like chlorophyll A and B, capture light. So these are the carotenoids and the xanthophylls. And actually they have two purposes. One purpose is to capture light, just like chlorophyll A and B, but if you look at the last bullet, it also says that they protect the chlorophylls from damage by stabilizing free radicals. Isn't it true? Don't you know a lot of people that eat like a lot of pumpkin, or they eat a lot of carrots? And the carrots and pumpkin have a lot of carotenoids. And you eat them because you think that you're being healthy and that it's protecting you. Well, there is protection, like there is protection in plants. The protection is they're preventing damage by stabilizing free radicals. So this is what beta carotene looks like. So when you eat a carrot, this is what the pigment looks like. So notice that there's a ring at each end of a carbon-hydrogen chain. Okay, so now let's talk about light. Okay, so remember, photosynthesis has two basic steps: the light capturing reactions and then the Calvin cycle. Well, in the light capturing reactions, chlorophyll is going to absorb the energy of light. And so the pigments are crucial in absorbing light. And what they're going to do with this high energy is they're going to work in clusters and actually start exciting electrons. And you already know where the excited electrons are coming from. They're coming from the splitting of the water molecule. In other words, the oxidation of H2O. Now, we already know that chlorophyll can absorb red light and blue light, and we know that it will not absorb green light because that's how we're seeing the color green. It's absorbing all light except for green. So notice here, blue light has very short wavelengths and red has longer wavelengths. And remember, there's an inverse relationship between wavelength and energy levels. Blue light can cause way more energy than red light. So you see that the blue light has taken electrons to a second stage energy level, whereas red light only takes it to a first stage. So several things can happen when we have excited electrons, and actually the pigments do absorb the light and they transfer it to the electrons. So one thing is the electrons get all excited and then nothing happens. Then they fall back down to ground level. They lose their energy in the form of heat or light. Now, photosynthesis only makes a mistake 2% of the time. 98% of the time, the light that is captured by excited electrons is used for photosynthesis. But when it isn't and it goes back down to ground state, this is called fluorescence. Fluorescence is emitting light because the excited electron was not used to make sugar. Okay, now we have two photosystems, and I'll be calling them, instead of photosystem, I'll say PS, PS1 and PS2. So the first thing that you note in the first bullet is that chlorophyll works in groups. So it's working in clusters, and what's happening is the pigments are capturing all of this light, and they're going to transfer it to the reaction center. And so we see, sometimes I see light as a pigment. The pigments are capturing light, and then they're going to transfer this light to a reaction center through something called resonance. When pigments in the antenna complex absorb light, they start to transfer it, because remember, the pigments are working together as clusters to the reaction center. And there in the reaction center is an electron. And this electron will become excited with all of the energy being transferred to that site. It says after transferring energy, electrons fall back to its ground state and energy is transferred to another photosystem. So let's take a look at the reaction center. So energy is released from electrons, and it can be emitted in the form of light, and that's fluorescence, or can give off heat. Or it says here we can excite an electron in a nearby pigment, or we can transfer this to an electron acceptor in a redox reaction. And in step 4, this is the beginning of photosynthesis. So those steps reflect what we see in figure 10.10, and I actually just really like figure 10.10 because I do feel in this case pictures are worth millions of words. In the first case on the left we have fluorescence. The electrons get all excited in the electron center or the reaction center, but no other reaction is taking place. So then the electron falls back down into ground state, and that energy is released as either light or heat. In the second frame, we have resonance. You know what resonance reminds me of? It's like the wave in a football field. We're not moving up and down when we raise our hands up and down. The next person is, and that's what's happening with resonance. pigments stay in place and they only transfer the energy. And notice right in the middle, that dark green center is the reaction center. And at the reaction center, this is where electrons from the splitting of the water molecule will become excited. So here you see the last scenario, and this is where photosynthesis is actually taking place. Here at the reaction center, an electron is excited, and when it's very excited and it's full of energy, there's an electron acceptor that will actually accept it. So this is a series of redox reactions. Remember oxidation and reduction? Remember, if you accept an electron, what is it? Is it oxidation or is it reduction? Yeah, you're right. It's reduction. We have something called the enhancement effect. And we're going to see a diagram to see exactly how it works. But in the end, what does it mean? It sort of means that two heads are better than one. We have two photosystems that comprise the light capturing reactions, photosystem 1 and 2. And they got their names because photosystem 1 was discovered first and photosystem 2 was discovered second. But in reality, which one comes first when we're capturing light? PS2 comes first and then PS1. So don't let the Roman numerals confuse you. Okay, so this is an example of the enhancement effect where we say two heads are better than one. So notice we have photosystem 2 and photosystem 1 indicated by dark red and light red. But notice when they work together, they actually capture more light energy and there's more oxygen production than if they were working separately. And that little extra bit is that yellow part that you see on the top of the third bar. So how does photosystem 2 work? And remember, it's the first one. PS2 comes first, then PS1. Remember where they're located? They're located in the chloroplast and they're associated with the grana and thylakoids. Remember, chlorophyll A and B, they're hooked in to the membrane. So it says here in photosystem 2, it's been studied, and we know the specifics because we've actually also used the purple sulfur bacteria so that we can understand the movement of electrons. So we understand that that electron acceptor at the very top, when the electrons become excited, it's another molecule that is accepting them or that is being reduced. And that molecule is a pigment called pheophytin. And pheophytin is going to take that electron and then transfer it or pass it onto an electron transport chain. And you guys already know what happens. Ultimately, in an electron transport chain, what is being produced? Yeah, you're right. It's ATP. Okay, so the electron transport chain means that high energy electrons are going through a series of oxidation and reduction steps. And at each of those steps, they're losing energy. But remember, energy can't be created nor destroyed. It can only be transferred or transformed. And so that energy will be transformed to actually ultimately produce ATP. Excuse me. I love this diagram. In this diagram, it shows photosystem 2. Photosystem 2 located in the thylakoid region of the chloroplast. Notice here we have high energy electrons. The electrons come from the splitting of the water molecule. Pheophytin at the very top is reduced. It's passing these electrons to an electron transport chain. And what is ultimately going to be produced? ATP. Photosystem 2 produces ATP through a proton motive force, the same way that ATP was produced in step 4 in cellular respiration. Now notice here that there's all different kinds of molecules involved in the production of ATP in PS2. We have plastoquinone and a cytochrome complex. But do you have to know this for the exam? And the answer is no. Do you need to be aware that there's a series of oxidation and reduction steps? Definitely yes. But the take-home message is that PS2 produces ATP through an electron transport chain. Okay, so as the electrons go down their electrochemical gradient, they're going to produce ATP. The proper technical way of saying all of this is the word photophosphorylation. So you know what, guys, let's pause and let's take a minute here. Now we're aware of 3 types of phosphorylation. And remember, this is important because energy can move through excited electrons or through phosphate groups. So there's substrate level phosphorylation, oxidative phosphorylation, and now there's photophosphorylation. In other words, the energy came from light, from photo, to actually create or phosphorylate ADP to produce or synthesize ATP. In this diagram here, you can see that actually we have an electron transport chain and a proton motive force, and that all of the protons, that's all of those hydrogens, are on one side of the membrane, and the only way that they can cross the membrane is through ATP synthase. So you should say to yourself, this looks very familiar to step 4 in cellular respiration. The only difference is that the protons were on the other side of the membrane. But when you stop and think about it, does it really matter? What really matters is the establishment of the electrochemical gradient. Okay, now please watch this video in your slide presentation, in the one that has no video to it. Okay, so here we can say that photosystem 2 is important. So what's happening in photosystem 2? If we stop and take a minute, this is where we split the water molecule. And why do we split it? Because we need the protons and the electrons. We don't need the oxygens. So water goes in and oxygen goes out. So what happens to the electrons? These are the ones that get excited. And how do they get excited? Because the pigments are capturing the energy from the sun and they're transferring it to reaction centers that ultimately excite these electrons. And because we're splitting water and releasing oxygen, this is called oxygenic photosynthesis. Now, those purple sulfur bacteria that are very unique and sort of rare, they don't use the splitting of the water molecule. We know from previous examples they're splitting hydrogen sulfide. So this is called anoxygenic photosynthesis. Now, will this be on the test? No, but it's just something to know, like in the back of your mind. Okay, so now we're going to move from photosystem 2 to photosystem 1. So what is the end product of photosystem 1? Photosystem 1 that also occurs in the chloroplast, in the thylakoids, its job is to produce energy. It's going to capture light energy and convert it to high energy electrons. And this brings us to our third electron carrier. Our third electron carrier is NADP. Now, I can tell here it's either NADP or NADPH. NADP is the oxidized form. NADPH is the reduced form. Remember, reduction means it is accepting electrons. So if it has high energy electrons, then the molecule itself has more energy than the oxidized form. So in photosystem 1, it passes excited electrons to the electron carrier NADP. Now, just like with photosystem 2 that happens first, is there are a series of other molecules in between. It isn't just straightforward. And it is important that you need to know that something is happening, but for this test, what's most important is that you know that the end product of photosystem 1 is excited electrons, and they're carried by the electron carrier NADP. So NADP is reduced to form NADPH. I love figure 10.14 because it reflects what's happening. Notice we can see that the pigments are working in clusters and they're working together. And all of that energy is transferred through resonance to the reaction center to excite electrons. And remember, these electrons come from the splitting of the water molecule. And in photosystem 1, the end product is excited electrons that are being carried by the electron carrier NADP. So NADP becomes reduced, and therefore we see it as NADPH. Remember, we always track the protons to see where the electrons are. So I love this summary table right here. I love this slide. It says electrons from photosystem 1 produce NADPH. Electrons from photosystem 2 produce ATP. So if you put them both together, like in the enhancement effect, PS1 and PS2 produce chemical energy in the form of ATP and NADPH. So now we've gone from light energy to chemical energy. So my candy bar is golden rays of sunshine. There's another video clip, and you can watch that in your other presentation that is loaded up on the Blackboard site. Okay, now let's talk about the Z-scheme. Okay, what is the Z-scheme? The Z-scheme is the enhancement effect. It is PS1 and PS2 working together. So both of those working together, we could call it the enhancement effect, or we can call it the Z-scheme. And because it looks like the letter Z, and that's why it's called the Z-scheme. Now, if you go through these forthcoming slides, they almost seem like a summary of what we've just discussed because they are. Now, in their example here in the book, they're going to use red and far red light, which is around 700 nanometers. And the truth is, I'm not going to go in detail over these, but I do want you to realize that the Z-scheme is the enhancement effect. And it is the combination of both photosystem 2 and photosystem 1 working together. PS1 plus PS2. Okay, so here it is. Notice it says P700. So 700 nanometers refers to red light. And notice here electrons will come in and excite at the reaction center, and PS2 will come first, and then PS1. So we know that ATP is going to be produced, and then we're going to have excited electrons. In figure 10.15, if you put your head sideways and you look at the figure over there, you can see the letter Z like Zorro, right? If you look sideways. So if you look sideways, you can see the letter Z. It's like the letter Z, like sleeping on its side. And this is why it's called the Z scheme. So notice you have excited electrons. Photosystem 2 comes first, and the end product is ATP. Then more light energy is captured by chlorophyll A and B working in clusters, and then it re-excites the electrons, and then for photosystem 1, notice the end product is 2 NADPHs. Okay, the Z scheme explains or is the enhancement effect. So that's the important thing that you need to know. It is PS2 and PS1 together. Okay, so here in figure 10.16, it's showing you how PS1 and PS2 work together. And another important thing to note is that it's happening in the chloroplast and it's happening at the thylakoid. So we have to have a membrane so that we can have this proton motive force or an electrochemical gradient. Now, your book talks about a non-cyclic and cyclic flow. For this class, we're not going to worry about it at all. There will not be any test questions about which one is which and what's the difference between them. So we're going to talk about the flow of electrons from the light capturing reactions now, and we're going to move those into step 2, the Calvin cycle. So I'm going to gloss over this. You will not need to know this for the exam. You do not need to know this. Okay. Okay, now this is an important point. I don't know if you've ever thought about this, but when the Earth formed, there was no oxygen gas in the atmosphere. There was water vapor, there was hydrogen, and there were likely other gases, but there was no oxygen gas. So where did oxygen come from? Oxygen came from the evolution of photosynthesis. And so oxygen is so important. And isn't it true, guys, if we didn't have oxygen, then we couldn't have cellular respiration. We wouldn't be making 25 ATPs in step 4. And if we couldn't make 25 ATPs in the electron transport chain, then we couldn't have huge multicellular organisms like myself. So is this like a real landmark in evolution of life here on Earth? And the answer is definitely yes. So the evolution of oxygen from photosynthesis is so important. So now we're going to move to the second part. We're going to move to the Calvin cycle, or the light independent reactions. Now, can the Calvin cycle happen in the presence of light? Sure it can. But does it need light? And the answer is no. So that's why we say it's light independent. It's not dependent on light. Okay, so CO2 has to get into the chloroplast. So how does it get in through the chloroplast? Well, there's like little mouths on the underside of the leaf. If I were to look at the underside of the leaf, lift up a leaf, there'd be like these little tiny mouths. And these little tiny mouths are taking in CO2 and releasing oxygen. Now, when oxygen is released, so is water vapor. And that is tricky business for a plant, because here all plants need water. Plants need water for photosynthesis, but they also need it for survival too, right? They need to keep hydrated. So plants are covered with this waxy coating called a cuticle, except for the underlayer of the leaf. And there we see these little mouths called stomata. And this is where we see CO2 going in and oxygen going out. Okay, so here's the waxy cuticle on the top, and that's why the water's beating up. And on the underside of the leaf is where the little mouths are, and this is where gas exchange takes place. Okay, so it says here open stomata when the mouths are open. When the mouths are open, CO2 will go into the leaf and oxygen will diffuse out. So these gases are diffusing against their concentration gradients. And remember, photosynthesis is an anabolic process. In other words, we're building sugar from CO2. Like that's someone's bad breath, right? So that's something really fascinating. In figure 10.19, you see the stomata that are found on the underside of the leaves. And they truly do look like little mouths. And these guard cells look like lips that are opening and closing the openings. And here you see oxygen and water vapor going out, and you see CO2 going in. And when CO2 enters the leaf, notice that within that spongy layer, it's going to go to the chloroplast. It's going to enter the chloroplast where it's needed to produce sugar. So the Calvin cycle is also also located in the chloroplast, and it's located in the stroma, the liquid or the matrix part of the chloroplast. And its job is to capture CO2. When we say capture CO2, the proper phase is carbon fixation. So it's going to pluck the CO2 from the air, and this is called fixing. And when it does, CO2 is an inorganic form of carbon, and then it's going to move to the organic form. So there's a 5-carbon compound called ribulose bisphosphate, and ribulose bisphosphate is the initial molecule that reacts with CO2. So we have 5 carbons in ribulose bisphosphate plus 1 in CO2. 5 plus 1 makes 6. So it says here, ecologically, the fixation of CO2 to ribulose bisphosphate is the most important chemical reaction on Earth. Can you believe someone saying something like that? Because ultimately it's the plants that harvest energy in a form that we can use. And at this point, CO2 becomes an organic molecule. And that is like beyond awesome. Okay, so here we see, here it says this is leading to the reduction of CO2. This experiment really doesn't help my own personal thinking, but it might help yours. So please go ahead and read about this experiment in your book. Okay, so the next thing I'd like to talk about is Rubisco. And it says here that CO2 fixation is done by an enzyme, and this enzyme is called ribulose 1,5-bisphosphate. But another way of saying it is just Rubisco. So what does Rubisco do? If I have my fingers here plucking CO2, then my fingers are Rubisco. They're the enzyme that connects CO2 to ribulose bisphosphate. Okay, so it's the most abundant enzyme in a leaf tissue, and it's found in all photosynthetic organisms because we need CO2 to make sugar. So here is what Rubisco looks like, and it has here one of 8 active sites. So every time you see an asterisk, you see that it's an active site which holds CO2. Now, one of the things about Rubisco, it says it's a slow enzyme, but this is what I think. It's like me, it's ancient. So remember, back in the day when photosynthesis evolved, and it only evolved once in the history of the earth, there was no oxygen. So back in the day it moved slow, and it moves slow now. And back in the day there wasn't any oxygen. But now that there's oxygen in the atmosphere, sometimes it gets confused. Just like I get confused, I guess because I'm ancient. So sometimes it can't tell the difference between CO2 and oxygen. Now, when it makes a mistake, is it making a big mistake? And the answer is yes. Because you guys know when there's oxygen, it's like cellular respiration. But because it's happening in plants, we call it photorespiration. So if it captures oxygen or plucks it and fixes it, then it's undoing the photosynthesis. So sometimes Rubisco cannot afford to make that mistake because it will undo what it's been doing. Okay, so if Rubisco by mistake plucks oxygen instead of CO2, then it's going to start breaking down sugar, and this is called photorespiration. It's going to undo photosynthesis. So this flow chart sort of describes what I've been saying. If Rubisco chooses correctly, it chooses CO2, then it will ultimately pluck CO2 that will go to the Calvin cycle to make sugar. But if by mistake it plucks or fixes oxygen, then instead of going to the Calvin cycle, it's going to start breaking down sugar into CO2. It's going to undo photosynthesis. So in hot, dry places, stomata are often closed. And if the stomata are open, we need exchange of gases, right? CO2 has to go in, and the plant has to get rid of as much oxygen as possible, especially so Rubisco won't make a mistake. But the thing is, when it opens up its stomata, it's also losing a lot of water. So let me give you one example of this. I went to Ecuador, and they used to call the jungle rainforest. And it's true, you would see a cloud hovering right there at the top of the trees. And I often thought, silly me, it was because we were at high elevation, like we were hugging the clouds or the mountains were hugging the clouds. But the truth is that the plant life was releasing so much water vapor that the water vapor was actually forming clouds. And that's why they're called cloud forests, because there's so much transpiration that water's being released that clouds are being formed. Okay, now here in the desert, it's hot and dry. So what's going to happen here? Well, plants fry, right? You know, so a desert plant, I used to think that they were one of the ugliest plants on the world, all desert plants, especially here in the Chihuahuan desert. But now that I'm an old lady and I understand how dangerous it is to carry out photosynthesis in a hot and dry place, now I appreciate a plant. And when I walk by, I always applaud and say, bravo, bravo, you're surviving. Because every time they open their stomata so that they can absorb CO2 and release oxygen, then they have to release water. And that could be the death of them, right? They could desiccate and wilt and die. Now, plants have two little tricks to live in hot, dry places. So I want to talk about these two little tricks. So the first trick is the C4 pathway. So what's happening in the C4 pathway? Where there's a division of labor between different cells in the leaf. So not all of the stomata are open at one time. Only some are, not all of them. So some of the cells open up their stomata and they capture CO2 and then they store it as a 4-carbon molecule. And that's why it's called a C4 pathway. Then they release it again as CO2 to other cells that are doing the Calvin cycle. So what's the little trick here? The little trick is there's a division of labor. So some cells have stomata and open them up, and other leaves do not. So that way the plant is more efficient and doesn't lose all of its water. So here are the details, and you don't have to know the details for the exam, but you have to know the idea of it. So there's a division of labor between cells and leaves. Some leaves open their stomata and some leaves do not. So some leaves open it up and they will capture CO2 and they fix it on to a 3-carbon molecule. So 3 carbon plus 1 is 4. 3 plus 1 is 4, so that's why it's called the C4 pathway. Okay, so then when the Calvin cycle is ready, then they'll re-release it as that 1 carbon as CO2. Okay, so it says here the C4 pathway does not replace the Calvin cycle. It's sort of like an intermediate step between carbon fixation and the Calvin cycle. So it's an additional fixation step. It's something there to make photosynthesis more efficient. So it says here that we have two types of cells in C4 plants, the mesophyll cells and the bundle sheath. So the mesophyll cells are actually carrying out or fixing CO2, and the bundle sheath cells are actually making the sugar. Okay, so this is the actual pathway, and for those of you who really enjoy plants, please take a look at it, but I want you to know the basic idea of it. So let me give you some C4 plants. Okay, so let's see. sugarcane, pineapple, a lot of the grasses. So, like when I was young and I wasn't doing much, just sitting around, people would go, "What are you doing?" or ask, "What are you doing?" And then people would say, "I'm watching the grass grow." But there's some places that you can actually watch the grass grow in one day. And in tropical regions, C4 plants are so efficient that they have measurable growth every single day. And for those of you who've been around sugarcane, you know what I'm talking about. So sugarcane can get to be like 6 feet, 8 feet tall, and it is a very hardy crop. Okay, so here you see the difference between C4 plants and C3 plants. So what are C4 plants? It's like the grasses and pineapple. And what are C3 plants? Isn't it true, like here in El Paso, if you want to have a vegetable garden, you have to tend to your plants every day. They're so delicate. And if they just get a little bit dry, then they wilt and sometimes they don't come back. So C3 plants are those vegetable plants that you have to sing to make them survive in a desert, in a hot, dry place. But C4 plants, the C4 plants like the yuccas, bear grass, all of those that you see in the desert, they have this little trick, this division of labor between mesophyll and bundle sheath cells. So here you see the two types of cells, the mesophyll cells and the bundle sheath. So it's a division of labor. And this way, they're very efficient. And this almost looks like a corn plant right here. It almost reminds me of the corn maze, right, that we have out in the outlying areas of town. So remember, why is it a C4 plant? Because the C3 molecule plus a 1 carbon makes 4 carbons. So the extra step is that CO2 is fixed and stored as a 4 carbon molecule and then gradually re-released to CO2 to move to the Calvin cycle in bundle sheath cells. There's another trick. Instead of a division of labor between cells, there's a division between night and day. So succulents like cactus, very succulent and fleshy plants, have a CAM pathway, a crassulacean acid metabolism pathway. They also store CO2 as a 4 carbon compound, but the way that they do this is they open their stomata at night, not during the day when it's hot and dry. So they only open up their stomata at night, and this is how they're able to survive in the desert. Now, because of this, we know that cactus don't grow very fast, but at least they don't die. So they're very slow growing, but they survive in hot, dry places. So notice here we have a saguaro cactus, and notice here the CO2, when the stomata are opened at night, capture or fix CO2, and they store it in this additional step as a 4 carbon compound. Then they re-release CO2 to the Calvin cycle during the day, and this is when sugar is being made. So it says here both C4 and CAM pathways function as CO2 pumps. So they minimize photorespiration and by regulating which cells or when the stomata will be open. So in C4 plants there's spatial separation or there's temporal separation. Spatial is a division of labor and temporal is day versus night. Okay, so what are the steps of the Calvin cycle that take place in the stroma found in the chloroplast? There's fixation, reduction, and regeneration. Do you have to know all 3 steps intimately? The answer is no, but do you have to know the idea of it? And the answer is yes. So we've been talking about fixation, and we've been talking about rubisco and ribulose bisphosphate. So we already know about fixation. So let's see if we can go on to steps 2 and 3, reduction and regeneration phases. Okay, so we know that the Calvin cycle is these 3 steps. So every turn of the Calvin cycle takes 1 CO2. So 3 turns makes this intermediate sugar called G3P, and 6 turns should make glucose. Okay, so what happens now? We're going to take all of the energy and all of the electrons and protons harvested in the light capturing phase, and now all of them will move to the Calvin cycle. CO2 will come into the plant leaf, and now we have all of the ingredients that we need to make sugar. Okay, I love figure 10.26 because you see the 3 phases: fixation, reduction, and regeneration. So in reduction, do we accept electrons or do we lose electrons? Yeah, you're right. We accept electrons. Now, sometimes when I think of the Calvin cycle, I think of my daughter. Now, why is it that I think of my daughter? Because when she was young, she used to brag that she was a high consumer. In other words, she was a high maintenance girl. And my husband always used to joke, look, you're so low maintenance. How is it that you could have a daughter that's so high maintenance and needs so many things? Now, I just want you to know, now that she's an older woman, she's not high maintenance. But the truth is I'm bringing this story up because the Calvin cycle is high maintenance, just like how my daughter used to be when she was young. So a lot of the energy, if you take a look at the steps, most of it, 5 G3Ps are used for regeneration, and only 1 G3P will be used to make sugar. So this is a high maintenance cycle, but the truth is this is how we make sugar. So how is photosynthesis regulated? Remember when we were talking about cellular respiration and all of the steps? Well, most of cellular respiration is regulated by feedback inhibition. But what really regulates photosynthesis? Well, the first thing is you have to have light for there to be photosynthesis. So light triggers photosynthesis. And high amounts of CO2 also trigger photosynthesis. And there are a lot of people who are growing high cash crops, and they'll even purchase these things called CO2 generators to actually produce a lot of CO2. Even though we know CO2 is a greenhouse gas, some people will actually try to give plants more CO2 so that they can produce more sugar. So the presence of CO2 actually triggers the fixation by the enzyme Rubisco. So what happens to the sugar that is produced by photosynthesis? Okay, so the Calvin cycle actually produces G3P. G3P is a 3-carbon sugar. G3 and 3 makes 6. 6 carbons is glucose. So we have this thing here called glucogenesis. So we can use G3P to actually synthesize glucose. Okay, now when we have an abundance of glucose or the abundance of sucrose, a disaccharide, then the plant will start making starch, and it will store it so when it's growing or maintaining itself, it can use its reserves for another time. So remember, it says here sucrose synthesis takes place in the cytosol. Do you guys remember where the cytosol is? Yeah, you're right. It's that place in between the nuclear membrane and the plasma membrane. That's where the organelles are found. That's the matrix. So it says here, virtually every carbon present in an organic molecule can be traced back to photosynthesis. So that means like all the carbon in me had to have come or be traced back to the formation of sugar from photosynthesis. Like that's awesome and amazing, right? Okay, that's the end of this chapter. Best of luck with understanding it. Bye.